Okay, we're gonna make a start now. So welcome everyone to our A Level AQA Biology session. In today's lesson, we are going to be looking at cell membranes, and it's the last lesson on cell membranes. We're going to be focusing on exam questions in today's lesson. Okay, so I've picked out common exam questions that you could be faced with in an exam. So I've gone through a lot of papers and I've picked out the exam questions which you can make flashcards for basically. Okay, the common exam questions which you can basically prepare for and you can go over them and you would be able to write the same sort of exam, same sort of answer in an exam. So we're going to get straight into it. So I'm going to give you guys an opportunity to have a go at the exam questions first. And then after a couple of minutes, I'm going to go through the answers and I'm going to show you where the marks are coming from on the mark scheme. So the first question is on the screen. It says, describe the process of facilitated diffusion and active transport. And it's worth 3 marks. Okay, so I want you guys to firstly have a go at it, and I want you guys to type in your answer into the Q&A. And then I'm going to go through it and focus on where the marks are coming from. Okay, if you're struggling with any of these questions, just let me know and we can go through them. So once you have an answer, let me know in the Q&A, and then I'm just going to start going through the answers in about a minute. Okay, just 30 more seconds, and then I'm going to start going through the answers. Okay, I'm going to start going through the answers now. So it says describe the process of facilitated diffusion and active transport. Okay, it's not asking you to compare them, it's just asking you to describe the process. So first of all, we've already looked at this a couple of lessons ago. Facilitated diffusion. So we know that there are two types of diffusion. There is simple diffusion and then there is facilitated diffusion. Now, the reason why we have facilitated diffusion is because we need to move things which require a carrier protein or a channel protein to move through. Okay, and these sort of proteins, they are only required when you have polar molecules or you have charged molecules or you have things which are not lipid soluble. Simple diffusion is used for stuff which is lipid soluble that can just diffuse across the phospholipid bilayer. Okay, so remember the plasma membrane which surrounds the cell membrane. Um, the plasma membrane is basically your membrane and it's made up of phospholipids. Okay, and that's why we call it the phospholipid bilayer. Things which can simply diffuse across the phospholipid bilayer, so that means just high concentration to low concentration, just through the membrane, that is simple diffusion. And that can only happen when you have lipid soluble substances. So fats, smaller fats, they can simply diffuse from outside the cell to inside the cell because they are lipid soluble. However, when you have facilitated diffusion, you normally require a transporter protein, such as a channel protein or a carrier protein, for this to happen. And this is normally the case for polar molecules or molecules which are charged. Okay, they are not lipid soluble. Okay, so how I would answer this question for facilitated diffusion, it's the diffusion of polar molecules down the concentration gradient via a carrier protein. Okay, so it's the diffusion of polar or non-lipid soluble, and whenever you're talking about this at A level standard, you have to talk about the concentration gradient. So it moves down the concentration gradient. So it goes down the concentration gradient, and facilitated diffusion, it happens through a channel protein or a carrier protein. So via a we'll say carrier protein. So that's the full-on definition for facilitated diffusion that you need to learn. For active transport is literally the movement against the concentration gradient via a carrier protein using ATP. So with active transport, when we looked at it, remember ATP binds onto the inside of the carrier protein, causing it to change shape and releasing the molecule on the other side. Okay, so it's the movement against the concentration gradient. Whenever you mention active transport, make sure you mention ATP. Okay, using ATP, and also mention what it's going through. So it's via a carrier protein. So this happens via a carrier protein. It doesn't happen via channel protein, because channel protein are normally only for water soluble substances. Yeah, channel proteins are those things that stem the whole length of the plasma membrane that goes through. Carrier proteins are literally they don't stem the whole of the, they basically have a weird shape like that, binds to the inside of it, changes shape, and releases it on the outside. Something like that. Channel proteins are like pores which fill up with water. All of this fill up with water. And anything which is water soluble, such as polar molecules or any ions, they dissolve in the water and they transport through to the other side. That's what a channel protein is compared to what a co-transfer carrier protein is. So if I show you the mock scheme, so again, common exam questions I've picked out here. Facilitated diffusion. It's the movement of polar or charged molecules or non-lipid soluble molecules down the concentration gradient. And you also have to say via what? Via your carrier or channel protein, or you could have said via a co-transporter protein. Active transport is the movement against the concentration gradient via a carrier protein. It's a carrier only, not a channel protein, using ATP. So whenever you're talking about active transport, mention ATP, mention the type of protein. Whenever you're talking about any form of transport, you have to mention what it's going through. So in terms of facilitated diffusion, it's going through a co-transporter protein. It's going through either a channel or carrier protein. Okay, active transport is only through a carrier protein and it uses ATP. Okay, so that was our first exam question. Our next exam question is here. It says explain why phospholipids can form a bilayer but triglycerides cannot. Okay, again, I'm going to give you guys a couple of minutes to start having a go at this, and then I'll go through the answers with you. Once you have an answer, you can let me know into the chat and I can have a look at it for you. Okay, just 30 more seconds and then I'm going to start going through it. Okay, I'm going to start going through it now. So it says explain why phospholipids can form a bilayer, but triglycerides cannot. So this is basically from our biological molecules in them. Now, we looked at the structure of phospholipids and triglycerides, and we saw with phospholipids, phospholipids have a phosphate group attached to the glycerol, and that phosphate group is charged, which means that it's hydrophilic. The fatty acids that are attached to the glycerol, the two fatty acids that are attached, they are hydrophobic. They will repel water. Okay. Now triglycerides, they don't have a phosphate group attached to them. Because they don't have a phosphate group attached to them, they don't have any hydrophilic part in them. Hydrophilic means that it can interact with water. Okay. Now, because there's a hydrophilic part in phospholipids, they are able to interact with water and then they can form a bilayer. So if we think about it, forms a bilayer such as this. Okay, this is a very simplified diagram. But what's happening over here is that in our plasma membrane, the phospholipids, they line up at the edge of the membrane. They form the edge of the membrane. And what happens is that this middle part over here, this part that we have in the center, this is hydrophobic. So it's hydrophobic. And this part at the end is hydrophilic, which means that it can interact with water. Now, because you have those two separate parts, one hydrophobic and one being hydrophilic, that means that you can interact with water and you can interact with fats as well. So triglycerides, they don't have the two aspects. They don't have a hydrophobic aspect and they don't have a hydrophilic, both of them together. And because they don't have both of them together, they're not suitable to form the bilayer that phospholipids can. The reason why they can form that bilayer is because the phosphates at the end, these circles, they will repel each other and they will line up against each other, whereas the fats can interact with each other and then they will line up in the middle. And that's why this structure spans the whole of the plasma membrane. And that's why substances which are fat soluble can just diffuse across. Okay. If they're not fat soluble, they have to go through a different means. So they have to go through either a carrier protein or a channel protein to move into the cell. Things that are only fat soluble can just simply diffuse across because of this hydrophobic fatty acid layer that we have in the plasma membrane. Okay, so the way to answer this question, why phospholipids can form a bilayer but triglycerides don't? Well, first of all, we know that phospholipids are both have hydrophobic and hydrophilic parts to them. Okay, so have both hydrophobic and hydrophilic parts to them. Okay, and because they have these two parts, they can interact with water. Okay, they can form a bilayer and can interact with water. So the phosphate group can interact with water. Okay, and then you have to say the converse of that. You have to say that triglycerides, they only have hydrophobic parts to them. Okay, so triglycerides have only hydrophobic parts. okay, which is the fatty acid tails that they have. Okay, so that would be your 3 marks. Okay, now if I show you the mark scheme, so it says phospholipids, both hydrophobic and hydrophilic, that would get you 1 mark. You could say that phospholipids are polar because they have a phosphate group attached to them. You could say that for your 2 marks, triglycerides, they are only hydrophobic or they are non-polar. Um, you accepts converse of that, triglycerides are not hydrophilic. You could also say for your 3rd mark, hydrophilic groups or phosphate groups, they attract the water. Okay, and they are present on either side of the layer. So if we think about this, we'll say that this is outside of cell facing outside, this is facing the inside of cell. Inside you're going to have fluids, so those phosphates can interact with the fluids, and outside you're going to have a bit of water, so the outside of your plasma membrane is going to interact with that water. Okay, so that's how you would answer that question. Okay, we're going to move on now again. This is our next question. Again, I'm going to give you guys 2 minutes to have a go at this one and then type in your answer into the chat once you're ready. And then I'm going to start going through it in 2 minutes. Again, this is a very common exam question. Okay, I think this came up in last year's paper as well. Okay, so have a go at it. I'll give you guys 2 minutes and then I'm going to start going through the answers. Okay, just 30 more seconds and then I'm going to start going through the answers. Okay, I'm going to go through the answers now. So it says in 1935, scientists suggested a model for the chemical structure of a cell surface membrane. Figure 2 shows the membrane structure the scientists suggested. Okay, so we're given that structure, and you can see that there are proteins on the outside, and then there's a sort of phospholipid bilayer on the inside that you can see. Now we need to give one similarity and 2 differences between the membrane structure shown in figure 2 and the fluid mosaic model of the membrane structure. Now, first of all, first thing that I can point out straight away, that both of them, the fluid mosaic model and this model, have a phospholipid bilayer. This structure that you can see over here is the phospholipid bilayer. So it's a similarity. I'm going to straight away put down that both of them have a phospholipid bilayer. Okay, now the 2 differences that we can have. Now, over here, we've got proteins on the outside. Okay, we've got proteins on the outside. Now, What else is present in your fluid mosaic model that is not present over here? Fluid mosaic model also has your channel and carrier proteins. In this case, you just have your phospholipid bilayer and the proteins are just on the outside. Okay, so you can say that difference one, fluid mosaic model has channel or carrier proteins present within the cell membrane. So fluid Okay, and we need another difference now. What else is present in the fluid mosaic model that is not present there? So when we looked in our first lesson on this topic, we looked at the structure of a cell membrane, and we saw that there were different components which was present within the plasma membrane. Okay, now some of those aspects, they were your glycoproteins and they were your glycolipids, your cholesterol. Those were all examples of stuff which were present in your plasma membrane. So in this case, another difference, in this example that we're shown, there are no glycoproteins and there are no glycolipids. Okay, so what I'm going to say, there are no glycoproteins or glycolipids present in this model. Okay. So that's what I would write down for 3 marks. Okay, if I show you the mark scheme. Okay, if we have a look at the mark scheme, it says both of them have a phospholipid bilayer. That's 1 mark. Both of them have acid or hydrophobic tails pointing inwards to each other. Another way of describing the phospholipid bilayer. Both of them have a phosphate, polar or hydrophilic heads pointing outwards. That's another thing that you could have said. Both of them have proteins. Okay, so those are some of the similarities that you could have stated. You could have also said for your differences, no channel proteins or carrier proteins, whereas your fluid mosaic does. Your cholesterol, glycoproteins, glycolipids, they are not present, whereas in the fluid mosaic model, they are present. Okay. Now, the next question, we're just going to move on. If you have any questions at any point, remember you can let me know. This is the next question. It says, compare and contrast the processes by which water and inorganic ions enter cells. Okay, so you need to compare and contrast, which means that you need to work out similarities and differences. So similarities and differences between the two processes which could occur. So again, I'm going to give you guys 2 minutes to start having a go at him, and then I'm going to go through it with you. Once you have an answer, you can let me know in the chat. Okay, I'm just going to give you guys 30 seconds and then I'm going to start going through it. Okay, I'm going to start going through the answers now. So it says compare and contrast the processes by which water and inorganic ions enter cells. So the way that you would answer this, so again, we're looking at water and then inorganic ions. Okay, so water, we know that it's going to move through osmosis and it's going to move down the concentration gradient. And we know that it's going to move through a channel protein because that's how water moves across a cell membrane or a plasma membrane. Ions, they can dissolve in the water and they can move across with the within the channel proteins. However, if you think about co-transport, in co-transport, we had sodium ions and they were moving out by active transport. So ions, they move against the concentration gradient by active transport. So if we start off with the similarities, both water and inorganic ions, if we have them together, mixed together, they can move down the concentration gradient through um, a channel protein in the plasma membrane. So both can move, they have the ability to move down the concentration gradient and both of them can move through channel proteins within the plasma membrane. So both can move through channel proteins. Okay, now contrast what happens with ions, they can also move by active transport against the concentration gradient. That's the contrast that you need to put in. So ions can also move against the concentration gradient via your active transport. Okay, if I show you the mock scheme for this, if I show you the, okay. So inorganic ions moved into cells by okay. So facilitated diffusion can only take place when you've got ions from a high concentration to a low concentration, and it can also happen through a channel protein or a carrier protein. Now, this question is saying you need to contrast a way between water and inorganic ions because you need to contrast a way that they are moving into the cell and it's talking about inorganic ions, they are basically relating the question to co-transport. And if you remember, in co-transport, sodium ions are actively transported out of the cell into your capillary, and that lowers the sodium ion concentration within the cell. Okay, so they can also move by active transport, going from a low concentration to a high concentration. The only reason we need to use it in this question, the only reason is because it says compare and contrast. So we need to look at similarities in the ways that they can move in. So the facilitated diffusion part that you're saying, that's absolutely correct. That's what the first two marks are for. They are both moving down the concentration gradient, and they can both move through channel proteins within the membrane. That's where the first two marks are coming from. This is your facilitated diffusion marks. Okay, and that's how you would normally see inorganic ions moving, facilitated diffusion. However, contrasting, we need another method compared to water. Water will never move by active transport. Water will only move by facilitated diffusion or osmosis. Okay. In contrast, over here, ions, they can also move via active transport. And you just need to think of the idea of co-transport in the process that we looked at. Sodium ions are actively transported out of your cell into the blood, and that moves by active transport from an area of low concentration to an area of high concentration. And that lowers the concentration of sodium ions within the cell. Okay, so what we are going to do is we're going to move on, and I'm going to go skip to a question that I had over here. So we're going to have a look at this question. Okay, and this will answer the sodium ion question that you had. Okay, so this is an example of a question that I found, and this is linking the process of co-transport into what happens. So in the diagram that we saw in our last lesson about co-transport, this is linking to the idea. So again, I'm going to give you guys 2 minutes to start having a go at this question, and then I'm going to start going through the answers. If you have an answer, you can let me know into the chat or the Q&A, and I'll go through them with you. Okay, just 30 more seconds, and then I'm going to start going through the answers. Okay, I'm going to start going through the answers now. So if we have a look at this question, it says the figure below shows the processes involved in absorbing amino acids into a capillary from the small intestine. It says name processes A, B, and C in each case, giving evidence for your answer. Now, if we have a look at process A first, now this is the process where once we've decreased the number of sodium ions in the small intestine, sodium ions are able to move from an area of high concentration in the lumen of the small intestine to a lower concentration in the wall of the small intestine. So that is facilitated diffusion through a transport protein, and no ATP is required. Okay, so remember, you've got a lower sodium ion concentration over here because sodium ions have been actively transported out of this wall of the small intestine. There's a high sodium ion concentration over here. And what's going to happen is that sodium ions are going to move across into this space and they're going to take a molecule of glucose with them or amino acid with them. So the process for A is going to be facilitated diffusion. And the reason why this is the case is because you've got a lower concentration of sodium ions in the wall of the small intestine. You're moving from an area of high concentration to an area of low concentration through a transport protein. No ATP is required. Okay, so there's no ATP required in this process. So that's evidence. No ATP required, plus through a carrier protein. Okay, now for part B, ATP is required. Because ATP is required, we know straight away is going to be active transport. Okay, because ATP required, that's your evidence. So ATP required. And this is the process where lower sodium ions are moving to an area of higher concentration over here. They're moving this way via active transport. And then finally, C is going to be from your interstitial space into the capillary. And that is simple diffusion. The reason why it's simple diffusion is because you're going to have a high glucose concentration over here in this place. And when we're moving into the capillary is carrying away all the glucose as it diffuses across. It's going across and it's carrying all the glucose away. So in the capillary, you're going to have a lower concentration of glucose at all times. So glucose is going to move from an area of high concentration to an area of lower concentration over here via simple diffusion. So it's going to be simple diffusion. And again, your reasoning is going to be no ATP is required. And because it's simple diffusion, you can also say that there is no transport protein also required. Okay. If I show you the mark scheme for this question, this is the mark scheme. So it says, in all cases, reject energy unless qualified. Always talk about ATP. This mark scheme is being very strict and saying reject energy. So the first part is facilitated diffusion. Transport protein needed, but no ATP required. B is an active transport because over there you can use energy, but ATP is required because over here you can see that ATP has been converted into ADP and phosphate. And then C is your simple diffusion because neither ATP nor a transport protein is required for the process to occur. Okay? So that's your mock scheme for this question. And this is a co-transport question that I found, which was a bit different from the original questions that we also